All right, Rajarshi. So what I'd like to talk to you about today is open notebook science, or making the scientific process as open as possible. And I'll be showing an application of how that can be useful for drug development. So first, a couple of definitions or an overview of what's going on. If you look at some of the trends that have been going on in the past few years, it's been moving from closed to open. And there are various ways that people make their research more open. Looking at this from the left-hand side, you have the traditional lab notebook, which is probably the most closed document in science that you have. Only the person who's doing the work, their supervisor, and possibly some colleagues ever get access to it. Once people leave a group, the lab notebook is, rarely accessed. So all of that information just gets lost. Then we have the traditional journal article, where it's open, but people have to pay for it. So you can get information, but it's more difficult for some people who don't have access to a good library at their institution. A very popular trend recently is the open access journal. And here it's actually free for people to access and to read. But again, here we're talking about the format is still as a traditional paper format. Okay, and that actually carries some information and doesn't carry other information. So today I'm basically going to be showing you open notebook science, where the idea is that you make the lab notebook public in as close to real time as possible, and that you have access to all of the raw data that the people in the group actually have access to. The particular research I'll be telling you about is the work that we're involved in making anti-malarial compounds. So this involves a collaboration between Rajarshi here. He actually has done docking for us. And we go ahead in my lab, I'm a synthetic organic chemist, so we make compounds. And then we ship them off to the Phil Rosenthal group at UCSF, who tests them for anti-malarial activity. And I'll give you some details about those results in a little bit. So when I talk about open notebook science, it's not one single technology that we use. We use blogs, we use a wiki. The lab notebook itself is actually on a wiki, but to share additional information, we might use Google Docs, ChemSpider for specific chemical information, mailing lists, and collaborative drug discovery, CDD. So it's really a host of technologies, and the vast majority of these technologies are free and hosted, so that if anyone wants to replicate what we're doing, they can do that overnight for free pretty much. So what's the point of this open notebook science? Well, it really comes down to, if you read an article and they're describing making a compound, and you click to the experimental section, you'll find that there's a lot of information that's missing. Sometimes you're lucky you can repeat the experiment, but other times there's information there that you'd like to see. So here, for example, in this blog post, I'm talking about Falcipain targets, and it says see experiment 150. So when I click on that link, it actually takes me to the lab notebook page on a wiki. And that actually looks very similar to a paper notebook, in the sense that we have a scheme. This is the Jiri reaction. And then we have an objective. And then we have all kinds of links. If people want further information, they can just click on the link and it'll give them everything that they need. So as an example, if we click on that Jiri adduct link, it takes us to the ChemSpider entry. So ChemSpider does a lot of things for us that is very nice. It calculates all kinds of properties. It gives us the SMILES, the INCHI. It gives us the molecular weight. And then this is actually a long list of things. It predicts log P, all kinds of properties that can be useful. And the other nice thing about ChemSpider is I can do a substructure search of my compounds in ChemSpider without having to worry about running software on my server. So that's very important for people who are not computational. So again, the idea here is that anyone who's reading that particular experiment will be able to tell exactly what happened. So one of the links is to the actual docking procedure that Rajarshi used, and here it has the actual library. It has as much information as necessary that somebody could repeat it, and it has actually the lists of the compounds in the order that he found them, just listed in a Google Doc here as a list of SMILES codes. So again, very, very helpful if we're talking. So I can make sure that if I'm communicating with somebody, who's related to the project, that we're really on the same page, and they can dig into information without having to ask me. There is also on that page a procedure section. The idea here is to write it up in such a way that you can copy and paste it in your article when you send it off for publication. We also use the JCamp format for most of our spectra. NMR spectra are very handy in this format. It's an open format that you can actually use JSpecView on your server, so that people who are visiting your site just through a simple browser don't need to download any more software to interactively expand your NMR spectrum. So that's something, sort of a pet peeve of mine, when you have the supplementary data of a paper and they have the NMR spectra in PDF format, you can't actually expand that. So you can't see if that wiggle, what it is, you can't measure J constants. But here, you actually, through your browser, you just drag across a peak of interest and it'll expand it as much as you want. And so it's very, very helpful. Even if you're not interested in open notebook science, this is definitely useful, within a group. And the other thing that we can do is push ChemSpider, more. If we actually have made compounds, we've characterized them, we're comfortable with what they are, we can actually store the NMR spectra, we can store images. The spectra are also in JCamp format, and it's the same thing. If you're on ChemSpider, you can expand any spectrum that you see as much as you want. So I think this is very, very promising for a number of organic chemists. ChemSpider also has a feature where you can get a predicted spectrum. So they're actually upgrading this feature right now. The upgraded feature should be available shortly. And this can be very handy. As you can imagine, you're talking with your students, you're trying to figure out what this compound is. It's nice that there's a button there. You can just hit it and it'll give you a predicted spectrum. And the most important section of any lab notebook is the log, because you can actually reproduce the rest of the experiment if you have the log. But if you don't have the log, it's completely worthless, because you don't know what happened and what people did. So this is the part of the document that I ask my students to have uploaded before the end of the day. They can upload data and other associated files a little bit later, but this has to be in so we know exactly what they did and observed. So again, I'm still on this same page that I started a few minutes ago. By the time you get to the conclusion, it says this Yugi product was obtained in 59% yield. Well, you actually don't have to take our word for it. You can actually go through the entire experiment, all the NMRs, all of the information, and you can figure out for yourself if that's how likely that is to be true. So there are various ways of accessing these experiments. Obviously, there's a table of contents, which looks just like a paper notebook. So that would be your first page on the paper notebook. And you can just click on any of these experiments. But you can also do Google searching. So we actually tag each one of our lab notebook pages with an INCHI and an INCHI key, so that people, if they're searching Google, can actually discover it. So if you're not familiar with the INCHI key, it's basically a string like this that is always the same length. So no matter how large your molecules are, Google will handle them properly. You can also use INCHIs, but the problem with INCHIs is, in fact, our Yugi products are a little bit too big. They're so long that Google tends to truncate them, so you can't really do a nice search with that. You can also use the Google Custom Search, again, another free service by Google, which allows you to specify which domains to search. So like right now, there's a useful chem custom search. So if you type something in there, it's going to search only my blogs, my wikis for my project. So those are some things that you can, again, free and hosted services. So how has this been working? Well, the idea is to communicate our information in real time and have people find the stuff that we put in there. So what's going on? So I have a site meter on every one of the pages of our blogs and wikis that tells us how many people are hitting it and what keywords they're using to actually find it. The vast majority are finding it through a Google search. So there are 4 different categories of types of searches that I've uncovered here. One of them is a specific compound. So if someone is looking for TFA and they want an NMR of it, that's the kind of search that they'll do. And searching Google is a reflex for a lot of us now. You basically do a quick search on Google. If you don't find what you're looking for, then you go to the other databases. So someone looking for an NMR of TFA, they hit it, they happen to find it on our site, they can use it immediately. People are also searching experimental conditions. So the kinetics of Bach deprotection. They happen to be in luck if they use that search term, because we've done extensive NMR monitoring of the loss of the Bach group. And so they can actually get in there and see very, very high level details. On a more general nature, we have the educational searches like organic chemistry quiz, how to make a poster in Second Life. So there are some more general things that people can find that are useful. There are some big picture searches like protease and malaria, lysosomal targets. So essentially, people are using this collection of sites exactly in the way that I want them to. They're finding the information that they're looking for in the searches. Now let me give you a very specific example of something that really in a traditional article you would not find. Someone searched for this term purification of phenylacetaldehyde on Google. And the first hit that they got was our experiment 37 on Useful Chem. And that happens to be done by James, one of my undergrad students. And indeed, that was his objective, to purify phenylacetaldehyde by distillation. Now, if you look at this experiment, there's a lot of useful stuff that people could find. You have his experimental setup here. All right, and you'll notice there's something a little bit strange about that setup I'll get to in a little bit. But you can see where the thermometer was positioned. You can see, pretty much how this thing was done. And there's an NMR of the results. This is actually before we use the JCamp format, so this is actually just an image. And you can see the NMR has its problems, but the majority of it is the compound. And of course, we have the log section. So it says exactly, what was done, what the temperature was, all right, and exactly how long it took to do all of this. Now, in the discussion, all right, it turns out that the phenylacetaldehyde is reported to boil at 195. However, the first fraction was collected at 162. The first fraction was the only fraction that the student saved. So the question is, there's actually a problem here. So it could either be because the reported boiling point is wrong, or it could be because we had a problem with our setup. There's a number of reasons, but the point is that you can actually see in detail, exactly where the problem might be. And also there were droplets floating in the flask, so that's always not a good sign. But so the bottom line is the distillate could not be used due to contamination, and it should be redone in our atmosphere, which was not actually part of that setup. So do we know that it's water? No. But is it possible that we're the only chemists ever to have tried to distill phenylacetaldehyde and had a problem? Maybe not. So that could be useful for someone who's trying to debug a reaction. And those are exactly the kinds of details that are not reported in traditional articles. Now, there's a lot of useful features of the way this is put together. We're using a wiki, and if you're not familiar with them, basically you can see the history of a page. So you never delete anything in a wiki. All you do is you replace it with another version, but every previous version is available and it's date timestamped. So you can actually see if someone changed their mind about something, or exactly when a conclusion was made. I'm just going to step through a few of these just to show you how this works. So on October 27, 2006, James basically started to record this experiment. And you can see that initially he actually wrote phenylacetaldehyde was purified. And that was something that was changed later on, obviously. And we can tell by comparing two versions exactly who changed it and how they changed it. The stuff in red are the deleted parts of the text, and the green is the part that got added. So basically, I looked at the information and I gave it my own analysis of what was going on, and that's where I started to give evidence as to why I thought there was a problem, linking to the published boiling point, things like that. Later on, all right, on June 23rd, my graduate student Khalid came in and actually put in a tag for phenylacetaldehyde. And then later on, if you'll remember that setup, I was asking if there's a problem. I had asked them did you run the water in the condenser, because there were no tubes connecting to the condenser. So the student then came back later and answered my question that there was water running in the tube. They were removed before the picture was taken. And that kind of interaction that I have with my student is one of the reasons I really like using a wiki to monitor what's going on, because I can give actually specific questions on specific parts of the experiment, and the student can respond, and we can have a conversation like that, which is very detailed. So this can be useful for many students. Shannon is another one of my undergrads, and there's a summary. And when she writes her report, she can actually link to each individual experiment that she performed, and she can specify her contribution to that specific experiment. So I think this is very handy for students who are trying to build a portfolio of what they can do, the kinds of observations they can make about their experiments. Having an open notebook can be very helpful. Now, I talked about Google is a good way to search for information, but the reality is if you've got 200 experiments, it's kind of tricky to compare them, right? So just like you would in a paper notebook, you make summaries of them so that you can compare. So we've been doing this Yugi reaction over and over again, and so it's convenient actually to put it in a Google Doc. We can put the different conditions that we used, we can put the researcher, and whether or not we got a precipitate. When we do this Yugi reaction, sometimes we actually get a precipitated pure product. And that's actually really useful. So we're trying to understand what conditions lead to that outcome. And so when we use this table, we can dig into this information. We also use other social software like YouTube. And this is very helpful. Instead of having the student write 3 paragraphs about every little detail of what they did, they can write something shorter and then take a video so that I can see where's the thermometer, what you call a reflux. Is that actually a reflux, or is it boiling off? Those are all things that it takes students time to actually understand exactly how to describe things. So video, I'm a huge fan of. Some interesting things happen when you use things like YouTube. One thing I found, I actually use it for my class as well, and I gave this talk about the NMR of an ester to talk about diastereotopic protons. And I got over 5,000 views, and it turns out that the majority, or I should say the highest viewers were older males, 45 to 55. So I would have assumed that would be the kind of thing pre-med students would be looking at, but you can never really know how your work is going to be done. And that's sort of the point of making it open. It can be used in ways that you hadn't imagined. All right, so we use other tools. A mailing list is very handy for inter-group collaboration. I know that Rajarshi and I use that, my students, to talk to each other. This is good actually for debugging things, where there's a lot of back and forth. And putting all these things together, we actually have a result that we'll be publishing soon. And the idea here is that we're targeting this enzyme Falcipain-2. And there are 2 sites on the enzyme that we've tried to dock against. So Rajarshi picked a V1 area and a V2 area. And I gave him a list of 71,000 compounds that we actually can make in my lab very quickly. They're all Yugi products. And we got some interesting results when we had the Rosenthal group test them. So all the ones for V1, and so these are rankings out of 71,000. All of the ones that have the V1 are actually do inhibit the enzyme on the micromolar range, and they do also inhibit the infection of the red blood cells, also in the micromolar range. Now, the ones that were docked against V2, they actually are also inhibitors of the enzyme, but they do not function to stop the infection of the red blood cells. And so actually 4 out of 5 have that behavior here, and all 4 have it here. We also sent them as controls just other Yugi products that we made that didn't show up high in the docking, and all of them, none of them actually did either. So this is kind of an interesting result. It shows us that something's going on. It's more complicated than it looks, though, because compounds that are known to kill the malarial parasite by blocking Falcipain-2 generally show a destructive food vacuole. And we don't see that in this case. So this may be a coincidence, but if it is a coincidence, it's a lot of data points. So we're going to continue to investigate that. And because this is done in the open, anyone who takes an interest can basically stop in and see exactly what's going on. And that's in fact one of the most useful things from this project is we found some great collaborators to participate. So information spread, I showed you the wiki, the blog, mailing lists. We can also use customized databases like CDD, where we can report our assay results. We can also put our compounds on e-crystals. Drexel now has a site. This is the University of Southampton project. You can put crystal structures in there, and people can search them, can make use of the data. And we can even go in Second Life. We can actually now display NMR spectra in Second Life, and we can talk to the spectra, and we can expand any area. So Second Life I'm not going to talk a lot about here, but it's something if you're interested come talk to me. We've been having meetings in Second Life, and you can actually show molecules, you can show spectra, you can interact with it. You can also take those JCamp files and apply macros to them. So Excel VBA is pretty convenient, where you can actually automatically calculate kinetics. And so that's another bonus of having your NMR spectra being in a JCamp format. So the other thing, again, you can do with Second Life is you can build these 3D molecules, and we have new tools now that Andy Lang developed in collaboration with me that make it really easy to be able to do that. You can also talk to reactions to see how they look in 3D. So on the bigger concept of Open Notebook Science, who else is doing this? Well, if you remember, I told you one of the advantages of my system is that it can be replicated by anyone immediately. And Gus Rosania at Michigan is one of these people who thought he might like to try it. And within a day or two, he had his own wiki. He's got 10 students now that are uploading their lab notebook every day on here. And so this is actually very exciting that the system actually can be used pretty easily. Other people are also doing a similar thing, but they're not necessarily using a wiki. Cameron Nayland from University of Southampton puts his notebook in real time, but he uses actually a modified blog engine. Now, the next steps in all of this is we'd like to go from, people analyzing the results, people putting in the information in the system. People are good at creative things, but they're not necessarily that good at paying attention to detail constantly. So we'd like to basically have the information available in a machine-readable format. And one of the things we've been doing lately is actually translating our log into a machine-readable format. All right? So here, basically, we use the INCHI key to specify this compound, exactly what happened, how long we waited, what we added when we added it. So this format can be scraped, can be ported into a database, and then you can do some interesting searching. So the last thing I'll tell you about is we have this robot that Mettler Toledo was kind enough to lend us in my lab, and we were able to do these 48 reactions in parallel, and we thought we would use this to actually do an optimization. So we took this Yugi reaction and we modified different parameters, concentration, the excess of some reagents, solvent, and we basically recorded the yield. And the other nice thing about using a robot, of course, is that it automatically records the log, right? Unfailingly, it will record exactly what it did at what point in time. And so we can get these XML files to basically dig in if something didn't go right. But the bottom line is that we were able to do this optimization of this reaction. And this is actually a 5D plot. Down this axis, we have concentration. So the higher the concentration, the higher the yield. The volume of the object is the actual yield. And as we move across here, different shapes correspond to different solvents. So we have acetonitrile and ethanol over here. They're pretty comparable yields. But THF, you see these little triangles, it goes down. And the highest yield of all is actually this blue sphere over here. So we managed to get the reaction from around 50% to about 68% by using this optimization. So the whole point behind this is, I showed you how useful it can be for people searching for information, and they happen, they're using Google, they happen to find our site. But ultimately, I really want to see more and more human-machine interactions and ultimately machine-machine interactions. There's no reason why a system like this can't evolve to having machines designing experiments, executing them, and actually analyzing them. And the only way that this is going to work on a global perspective is if all the information is free and open to be accessed, which is probably the main reason I'm a big supporter of open science. It's just, I think it's a more efficient way to get things done. And conclusion, pretty simple. Communicate first. So I've talked about standards after the fact, right? The most important thing is that people can understand what we did in the lab. So don't worry so much about standards before putting your stuff out there. Once you have your information out there, then start to look at standardization, like what we're doing with the logs, converting them to machine-readable format. And in a Science 2.0 world, redundancy is king. So it used to be you'd want to put all of your information in one paper and then have everyone cite that paper. Now actually, because things are so distributed, it makes more sense to take your data and to duplicate it in as many different databases as you can, because you don't know how people are going to find it. And this doesn't stop you from publishing your results in a regular journal. In fact, we're going to be publishing this optimization in the Journal of Visualized Experiments in the next couple of weeks, which should come out. It doesn't stop you from doing that, but it gives you the advantage of sharing what you're doing in real time immediately. And that's basically it.
Thank you.